Hello guys, this is Dr. Ramya from Team MDS CONQUER. In this 32 series, we are going to study some important points on the topic of protein metabolism. Point number 1. Gout is primarily a disorder of purine metabolism. Number 2. Uric acid is the final breakdown product of purine degradation in humans. Number 3. Primary gout results from either increased production or impaired excretion of metabolic end products of purine metabolism, that is the uric acid. Number 4. Urea is mainly formed in liver and to small extent in brain. Point number 5. Sources of carbon and nitrogen in urea or methane are carbamide, citrulline, arginosuccinate and arginine. Number 6. Threonine, lysine, phenylalanine, methionine, isoleucine and leucine, tryptophan, valine, arginine and histidine are nutritionally essential amino acids, whereas arginine and histidine are semi-essential amino acids. Among them, arginine is more essential than histidine. Point number 9. Hemoglobin is an allosteric protein. Allosteric protein is the one in which binding of a ligand to one side affects the binding properties of another site on the same protein. Phosphofructokinase is an allosteric enzyme. The allosteric enzyme is the one in which binding of a modulator to the enzyme induces a conformational change in the enzyme, making it more active or less active. Point number 13. Proteins are responsible for maintaining the structural strength of cells, that is the cytoskeleton and tissues. Point number 14. Collagen is the most abundant of the fibrous proteins. Other prominent fibrous proteins include keratin and myosin. Proteoglycans are made up of sugar and amino acids. Point number 17. Glycosaminoglycans hold large amount of water, thereby producing gel-like matrix that forms the basis of body's ground substance and lubricating to other structures. Number 18. Aspartic acid migrates fastest on chromatography on carboxymethylcellulose medium. Number 19. An alpha helix of a protein is most likely to be disrupted if a missense mutation introduces the aspartic acid within the alpha helical structure. Number 20. Aspartic acid, being a charged amino acid, disrupts the coil spiral structure. Number 21. Convergence is an evolutionary process by which distinctly unrelated organisms like protein acquire morphologically similar traits, whereas divergence produces different traits in related to the same ancestor organisms. Point number 23. Triple bonds are found between cytosine and guanine base pairs, whereas double bonds are found between adenosine and tyrosine base pairs. Folding of a protein chain is due to hydrogen bonds. The hydrophobic weak interactions are predominantly important for proteins stabilizing the conformation, whereas hydrophobic residues are largely buried in the protein interior away from water, and the number of hydrogen bonds and ionic interactions within the protein is maximized, thus reducing the number of hydrogen bonding and ionic groups that are not paired with the suitable partner. Number 27. Glutamine is the sole form of transport of ammonia in brain. This is an amide of glutamic acid which is formed from alpha ketoglutarate. Number 29. Alanine leucine is a conservative mutator. The secretory proteins and transport proteins are synthesized in ribosome attached to the endoplasmic reticulum, whereas proteins which have to be delivered in cytosol are synthesized in free polyribosomes. Point number 32. Chaperones are the proteins that play a role in the assembly or proper folding of other proteins without themselves being component of the latter. That's it for today's 32 series, guys. Hope you all note down these points, revise them well. All the best. Thank you. Signing off, Ramya, Team MDS CONQUER.